Pero volver a preguntar a mí me encantó mucho cuando me estaba haciendo. No hay que hacer investigaciones de
Yo lo voy a poner una regla. Estoy cansado de ser pie, ¿verdad?
Yo estoy buscando como que mis universidades de interés que sean como que solo grad school, que no tengan un under porque no quiero TA. Como que yo no estaría en contra de ser TA porque no pienso que es horrible, pero pienso que como que mi meta no es ser profesor, mi meta es como que tener mi research o estar en la industria, como que no como profesor. So, como que TA no me trae mucho a la experiencia que yo quiero tener.
Y yo quiero ser profesor.
Y enseñar es bonito,
Es diferente.
uno más de los
It's recording, so all your conversations. So when people go online, they're like, "What is this?" All right. So yes. Good morning, everyone. What we're going to do today is continue our discussions of metal compounds as they are applied for anti-cancer purposes. And so, in our last discussion, we focused on platinum because platinum actually still is the lead metal when it comes to anti-cancer regimens. And so what you see as far as what we'll talk about in subsequent lectures is other metals that people are still exploring, but many of these have not actually translated to clinical use. And so today we're going to focus on two different metals. And the reason for this is that they are on the two ends of the spectrum of hard, soft, acid base. We're going to look at titanium, which is a hard metal, and we're going to look at gold, which is a soft metal. And so we're going to see a bit about what properties of these metals lend themselves for potential use as an anti-cancer agent. All right, so we're going to first focus on titanium. So let's get a little bit of context about this particular metal. And so in our environment, titanium is quite abundant. In fact, we come across it every single day, right? So you may not realize it, but if you have white toothpaste, well, there's titanium in there, titanium dioxide. Sunscreen, there's a lot of titanium dioxide nanoparticles. White paint has a lot of titanium dioxide. And so a large portion of titanium is located where in our bodies? Do you guys know? It's located in our lungs because we breathe in a lot of these particles, okay? And so generally speaking, the metal is not believed to be something that's highly toxic, so and it's known to be fairly biocompatible. Well, in the environment, it can exist in a range of oxidation states. I have listed here from down to -1 up to +4. And so biologically speaking, it's the +4 that is the most relevant, but you do see +3 to some extent used, but more used in the chemical setting because it's something that is redox active. And so the most stable and most common oxidation state is +4, and that's because we are in an oxidizing atmosphere. So that favors titanium-4. The most stable form of titanium-4 is what I have already mentioned, is titanium dioxide. And so we commonly come across it as in its amorphous form, but it can crystallize into these highly stable forms, rutile, brookite, and anatase. And I'm going to talk a little bit about that. Some of these crystalline forms lend themselves for potential use for an anti-cancer application. So even within the reducing environment of cells, right? So that's where we've talked about how iron, because it can flux between iron 2 and iron 3, that's an important feature, the reducing environment of cells. Well, in the case of titanium, titanium will basically locked as titanium-4. And that's because there's no biological reducing agent that has the capability to reduce that metal from 4 to 3. So maybe one can design a compound of titanium-4 that, because of the ligation, can fine-tune the redox property of the metal, enabling it to be potentially something you might be able to reduce. But generally speaking, titanium-4 would be locked as that oxidation state. So as titanium-4, let's talk a little bit about the chemistry of it. So that is a D0 metal ion. So thinking back to what we talked about at the beginning of the semester, that's the electron configuration of it. So that means it is going to be diamagnetic. And also, another really important property that we discussed was crystal field stabilization energy. We talked about how that particular parameter can be a thermodynamic drive for the stabilization of electrons coming from the d orbital of the metals as it enters into a coordination fields. And in this case, an octahedral field is normally what we look at as our point of comparison. But titanium-4 doesn't have d orbital electrons. So obviously it's not going to benefit from any sort of crystal field stabilization energy. So because of that lack of that stabilization energy, you would expect that titanium-4 compounds would be quite labile to ligand exchange. And that can be true, but because of its high oxidation state, that high oxidation state decreases that relative lability of the ligand exchange. So here I provide you a table of some of the coordination numbers that have been identified for titanium and the examples of these. But generally speaking, what you will come across normally is a coordination number of 6, and you do have a coordination number of 4. And I'll show you in a moment a really important titanium compound in terms of an anti-cancer application. But the most common form is coordination number 6, and you're going to get some variation of an octahedral geometry, depending on the ligation. Now, if you explore the bioinorganic chemistry of titanium, so we already know it's a hard Lewis acid. So we can start making predictions as to what is going to be able to bind that particular metal. So here are some moieties that you come across biologically that do have metal binding capability for titanium. So you can expect something like an amine, nitrogen, carboxylate, oxygens. Even we've talked about how that can even serve as a bidentate ligand, and it does for titanium. Here I'm just showing you the monodentate modalities. You have amino acids, obviously, that do contain R groups that have the carboxylate. You have catechol. And so catechol is a common feature that we've already seen in siderophores. It's good for titanium. And so these are some really common groups that would bind titanium. So if you want to look a little bit more extensively, so here, not so easy to see, but the number of anions present in our blood that have titanium binding capability. You also have a number of siderophores that they're not necessarily designed by bacteria for titanium binding, but in terms of exploring the coordination chemistry of this metal, have very high affinity for titanium. And that makes sense because of the nature of these type of chelators. So one of the really important feature about titanium coordination chemistry is that it can mimic that of iron 3. And so what do I mean by that? Well, it can bind to many of the same type of ligands as iron 3 can, biological ligands. So here's just a comparison in terms of coordination to the catechol moiety. And so what we're looking at here are the type of coordinations that can happen with a catechol group bound to iron and bound to titanium. And so what we're seeing here is a difference of species where you have either a 1 metal, 1 ligand ligation, 1 metal, 2 ligands, or 1 metal, 3 ligands. Now, so that can happen for both of these metals. But when you look at it in terms of their behavior in solution, and that's what we're seeing in the plots to the right, we're looking at similarities of these type of species existing in solution. So we're looking at the pH-dependent aqueous speciation. But there is quite a bit of a difference in terms of what is more prevalent for one metal or another. And this falls in line with their relative Lewis acidity, where if you're looking at it from the context of the aqueous environment, titanium would be much more Lewis acidic. And that's something that's relative term. And so when we're looking at that property, we're focusing more on the ability to bind to, let's say, a hydroxide anion as a measure of that, the strength of that interaction. And so that would be higher for titanium than it would actually be for iron. And so you see that reflected in terms of this pH-dependent profile, where you get this 3-to-1 species that is prevalent even at a pH of 6. We're talking about micromolar concentrations of metal and ligand, but that type of species doesn't become prevalent for iron until you get to about a pH of 9. Okay, so same type of speciation, but not necessarily in the same pH-dependent fashion. All right, another really important thing to allude to what I've already mentioned about the redox properties of titanium. So titanium-4 as it correlates with iron. So these can bind to the same type of ligands. But what has been established, so this was a correlation that was done in this study here, but they were basically looking at the redox potential of titanium versus iron 3. So titanium-4 versus iron 3. And what's generally observed is that, let's just look at one example, this ligand here, which is actually a hexadentate ligand. you get something that is a reduction potential for iron that's between -300, -400 millivoltage per with respect to the NHE, right? That's the standard comparison. But then you look at it for titanium and you have something that's closer to -700. So the correlation is that for the same type of ligation and the same type of pH condition, you typically are going to get a much more negative reduction potential for titanium-4 than you would for iron-3. And that would typically explain why it would be harder to reduce titanium-4 relative to iron-3. All right, so another important feature of this metal is that it is hydrolysis prone. So we talked about this previously about the classes of transition metals that have a Bronston-Lowry acidic property. So that is the ability to release a proton. And so in this context, how do we measure that? Well, we measure that in the perspective of water molecules bound to the metal, so aqua ligands, and the release of protons as a consequence of that. So when we looked at that, what I mentioned to you was that metals that are oxidation state of 4+ or higher tend to be very prone to this. And so titanium falls really much in line with this. And in fact, you put titanium-4 in an aqueous condition in the absence of a chelator, what you're going to get is extensive hydrolysis, and its thermodynamic drive is the formation of, or its thermodynamic sink is the formation of titanium dioxide or some sort of titanium oxide species. So focusing on that particular material, okay, which is quite insoluble, if you were to try to predict solubility of titanium, so here's just an example of a titanium oxide species. If you were to try to predict solubility of this metal, you get about a femtomolar amount. So very low solubility of titanium. And if you were thinking about this in the context of, as I've put it for like even iron, it falls in line with something that would be quite unavailable. But it's observed that the combination of small molecules, such as citric acid or even tunachromes are like siderophores, but they're used by algae for binding different metals. Or proteins like transferrin, we know it has a metal binding site, or something like albumin, which also has distinct metal binding sites. These type of biomolecules that can serve as chelators have been shown to effectively increase the solubility of titanium. So you can get quite high concentration of titanium, and it's been observed in human blood that you even have as high as nanomolar amount of titanium. So this is particularly true for people that have implants that contain titanium. All right, so once you get into this level, there's a possibility of titanium becoming bioavailable. And I'm a little cautious about saying that because titanium in the human body is not known to be an essential metal, but it's known in general that the metal has bioactive properties. All right, so let's talk a little about titanium in the body, in the circulatory system. And it's been observed that titanium that is released into the blood will be captured by transferrin. So we talked about transferrin as a protein that is responsible for the biodistribution of iron in our bodies. And so it also has been observed to be able to pick up this titanium. The question is, and it's still something that people are addressing, what does it do with that metal, if anything? You do see some correlation between transferrin binding and the titanium that's distributed to the body. But again, no property of titanium with respect to the body itself. So here is a structure of how titanium can coordinate to this protein. So previously we looked at the iron coordination to this protein, and we identified the modality of how that metal is bound. So we saw 4 amino acids and a synergistic anion carbonate serving as to facilitate the binding of that metal. Well, in this particular study here, it was observed that titanium can bind to the metal binding site, the iron metal binding site. But here, the model that was observed was something that was similar but not identical, where we have the coordination of 2 tyrosines, we have the coordination of the bicarbonate synergistic anion, but very interestingly, the coordination of another anion, citric acid, or in this case, citrate at the pH of 7.4, that helps to facilitate metal binding to this protein. So that's a significant finding just because it shows a distinction with how iron binds, but more importantly, in this form of coordination, this would actually be an open confirmation for how the metal binds. All right, so this shows you similarities, but also the important differences between how these two metals can coordinate to the protein. And that's, again, a reflection of just the subtle differences in the Lewis acidity of these two metals. All right, so as far as what's proposed as a delivery of the titanium in terms of the transport and transfer, much of this is still under exploration because it's not clear its ultimate fate as how it might actually get released into the cellular environment. It's known that the titanium can be released into the cytoplasm, but how that actually happens is not so clear. Because in the iron case, as we've explored, the iron metal can undergo a reduction process. And that reduction process is critical when we're talking about the endosome, where for iron, that reduction from iron 3 to iron 2 is critical for the iron to be released through the divalent transporter. But in the case of titanium, that's not clear because the metal itself will not get reduced. It certainly won't get reduced to titanium 3 and even will not get reduced further to titanium 2. So it's not exactly clear how ultimately that metal will enter into the cytoplasm. So this is just sort of a visual of a proposed route, but this is actually not something that has been established. It's something that people are still trying to explore. And not just for titanium, for other metals that are not redox active, that are believed to be delivered into the cellular environment by transferring, still that process is unclear. Maybe there's some sort of ionophore that can remove the metal from the protein and then deliver it out of the endosome. But that's still something that's under investigation. All right, so let's look at, in general, the biomedical application of titanium. So we're leading towards this anti-cancer, but let's put this a little bit into perspective because titanium is effectively used in the prosthetics field, and so, or in dental field. And so because it has a lot of important properties that enable its use for implants. So a really important property is this one of osseointegration, that you can have bone develop on top of the surface of the material. So that's an important feature. Also, it's been shown that it is biocompatible. So a measure of this biocompatibility is that you can have a layer of biomolecules forming on it or actually being associated with it, proteins or small molecules. And there are really few examples of an allergic reaction to the presence of this metal. And it's proposed that some of the allergic actions that people have identified might be more due to a decomposition of material as opposed to the actual metal itself directly triggering this. But another thing that's observed in terms of its use in implants is that some amount of leaching can happen. It's not clear what triggers that. If it's something that is a sort of a transient thing that maybe some amount of material on the surface gets released, but that metal that is released has been shown to be captured by serum transferrin. And it can even drive the levels of titanium to even into the low micromolar levels. Remember I reported nanomolar amounts, you can get even low micromolar amounts in this kind of context. It's also observed that titanium has an antibacterial property. And so here, it's not entirely clear when we're looking in terms of small molecules, what is the general behavior of this, although some indications suggest that ligand association is important for the formation of titanium as a titanyl ion. So as a titanyl ion is a titanium oxide, which has a unit is 2+. And so there's some, they attribute some of the properties to the formation of this particular species, but it's not exactly clear why that is the case. But it's also been observed that titanium dioxide nanoparticles, so this can be in the rutile form and the anatase form, because of their semiconductor property. Okay, so that's something that I believe you were familiar with, and Alex will be familiar with in inorganic chemistry. Because of its semiconductor property, depending on the source of energy, whether it's UV or some sort of other thermal energy, you can excite the electrons within this material. And in fact, you can photocatalytically activate titanium dioxide nanoparticles so that they can kill bacteria. So it's believed that part of the mechanism of action of titanium dioxide nanoparticles is a disruption of the cell membrane. You disrupt the membrane, you can cause a lysis, which means you break the membrane, and that can cause for leakage and obviously ultimately into cell death. But in terms of a true clinical use of titanium, for anti-cancer properties, so for antibacterial properties, not known, but you do see a lot in the materials field, people layering things with titanium for this possible application. All right, so that leads into the properties of titanium that lend themselves for potential anti-cancer use. So something that I mentioned to you already is that titanium in general is prevalent in our body and is not known to be something that's toxic. But interestingly enough, depending on the coordination chemistry, you can fine-tune the property of titanium so that it can become cytotoxic or induce toxicity in cells. And so following the discovery of cisplatin, this was in the late 60s, the 70s, people became very interested in looking at other metals that can have a therapeutic property. And titanium was one of those metals that people started to champion. And first of all, you know, as I explained to you previously, one of the drives in terms of drug discovery was more of an empirical drive. Let's look at general features of a given, let's say in this case, metal and metal ligand compound, and try to see what bioactive property they have. So this led to the discovery of something like titanium chloride and even butyrotitane. These are two titanium-4 compounds. And so initially people were interested in these because they approximated cisplatin. So they look very different, obviously, from cisplatin, but they have that feature of this, in the case of titanium dichloride, of that cis arrangement of chloro groups, and in the case of butyrotitane, the cis arrangement of these ethanolate groups that can dissociate. So people are curious about, okay, well, if that exists, what is going to be the mechanism of action of these particular compounds? So what was observed in the laboratory setting was that these compounds do have a wide spectrum of effect, meaning that they can have a cytotoxic property across different types of cancer cell lines. And also, very importantly, was a lack of cross-resistance with cisplatin. So in our previous lecture, we talked about how there is many different lines of acquired resistance that cells can develop against cisplatin. And so those cells that become cisplatin resistant, these kind of titanium compounds were able to show activity against them, meaning that there was no cross-resistance. So these are some of the features. That in fact, the chloro groups and the ethanol groups on these compounds do in fact, when you put them into water, will be, or they're very labile, they will be released pretty quickly. The thing is that these two compounds did advance to clinical trials. Titanium dichloride made it to phase 2, butyrotitane made it to phase 1, but they didn't advance further. And so the reason why they didn't advance further is not because of any toxicity, it was because of the lack of much activity. So they were not efficacious. And the reason for that is believed to be associated with the poor aqueous chemistry, or the lack of stability of these compounds, where their thermodynamic sink is the formation of titanium dioxide, and titanium dioxide, this amorphous form, is not known to be something that has any activity. It's pretty inert. So the loss of the quick loss of these ligands and the formation of titanium dioxide is basically a dead end. But still, there's the curiosity about what about the metal that was observed to have an actual cytotoxic effect. What contributed to that? So you have a lot of studies over the years where people have tried to really understand the behavior of the metal and also tried to understand the coordination chemistry to improve it, to overcome this really common feature of titanium, which is that they can be quite unstable. Compounds can be quite unstable in an aqueous environment. All right, so here I'm presenting some information with regards to the understanding of the mechanism of action. And so you have to be a little cautious of how to interpret some of this, because you get a lot of information that are cell line dependent, but a general trend here that we're going to look at. So we're looking at an ovarian cell line A2780, and there are two forms that we're looking at. The normal form of that cell line, and then a form of it that has been mutated so that it's become cisplatin resistant, so A2780CP. And so you're trying to probe the activity of titanium dichloride in this study, and you're looking at IC50 values, so that number is kind of hard to see. It comes from the figure stuff that's kind of hard to see in general. So you look at IC50 numbers, and you're trying to look at the activity of the titanium dichloride and also the cisplatin against the cell line and a few different variations of it, the cisplatin resistant one. And so a really important parameter that you're trying to look at is what is called the resistance factor. Okay, so the resistance factor, and we'll establish this in the context of cisplatin. So against the regular cell line, A2780, you have an IC50 value that's 4 micromolar. Okay, so we're going to put this right here, 4 micromolar. All right, but then with respect to the cisplatin resistant mutant form of it, you have something that's now I believe that's 51 micromolar. So the IC50 value has gone up. Okay, the higher the number that is, the less potent it is, so less active. And so the resistance factor is basically that the IC50 value against the CP form of the cell line divided by the IC50 value against the regular cell line, and you get a value that's 12.7. So there's a resistance, and that's expected because that cell line is resistant against that compound. But then if you look at titanium dichloride, you have a resistance factor that's 1.47. So you don't have the resistance. That's why it says it lacks the cross resistance that you would have with the cisplatin. So that suggests that the compound is operating via a different mechanism of action. But, so that's great. And many people in that time were, showing that as a really important thing. It's why this compound was able to really go quickly to clinical trials. But if you look closely at a lot of this older literature, you will see that titanium dichloride was never that great. You look at this number, you're talking about 570 micromolar IC50, right? 570 micromolar. That today, when people make compounds, that's something that will most likely be rejected as far as worthy of going further because it's a really high value. But at that time, different setting, right? We're being very exploratory. We're trying to see what properties. And maybe you might consider moving ahead just for the curiosity as to why is there a lack of cross resistance, and maybe understand mechanisms of action to try to improve. But certainly that number is not something that's very impressive. All right. So what were the general properties observed for titanium dichloride? It's observed to be able to induce arrest of the cell cycle, and it was observed in the late S and the early G2 phase. So these are phases where you are basically inhibiting replication of cells. There was also observed that titanium can form DNA adducts, or at least it was observed that the titanium can actually localize in the nucleus. It can induce apoptotic cell death. Well, apoptosis is actually a programmed cell death, and it has the capability of doing this, and to do so at any phase of the cycle. Also observed that it can induce DNA strand breaks. Okay, so the mechanism of that, I'm going to talk about that in a moment. And it also leads to accumulation of P53. So P53 is an important protein to try to rescue DNA synthesis and replication. And consequently, if it is affecting DNA, it makes sense you get the accumulation of this particular protein. All right, so let's look at what we observe here. So what we're looking at is a gel that we're focusing on DNA. And we're looking at what happens to a DNA strand as you have different concentrations of titanium dichloride. So we have this band that's up here, basically not really move far through the actual gel. And so you're looking at here at 10 micromolar. So this is 10 micromolar, if you can't see that. This is 100 micromolar, and this is 1 millimolar. Okay, all right. So we see a dose response that when you get to a high concentration of the compound, you start to see the formation of these bands. Okay, what does that mean? Well, that implies that you can structurally be changing the DNA, and that's believed to be the case, but you're also forming fragments. And so those fragments are able to move further through the actual gel itself. But you really require high concentration, I already mentioned. So 100 micromolar is when you start to see formation of fragments, and when you go 1 millimolar, which is super high, you get a lot of fragments forming. And so, also observing the study here, and just try to clear this up so you can see this a little better, is the amount of titanium present in DNA, and you see this in a time-dependent fashion, that you have an increase of titanium over time. That's after 15 hours of treating these cells with the metal. And then you get the over time, but also in terms of a dose response, same concentration range of the expression or the accumulation of P53, okay? And you see that in terms of time as well, up to 12 hours, you see it increasing, and then it starts to decrease after that point. All right, so you see the effect that the metal can have in terms of DNA. There's another very interesting study, which is an electron spectroscopy image, where you're basically tracking the metal. And so this is a really nice way of being able to see where is this metal going. So here they were looking at the presence of the titanium in the hepatocyte cells. Okay, so using titanosyne dichloride and trying to see where it localized. So here's data for the titanium localizing in the nucleus. And as far as them being able to track elements, well, they also tracked phosphorus, and they saw a co-localization of the signal for titanium and the signal for phosphorus. So this suggests that the titanium, if it's localizing in nucleus, is localizing in a region where there's phosphorus, likely that is interacting with DNA having a phosphate backbone. And it's also observed that the titanium was localizing in lysosome, although not very clear if there's any actual activity associated with the lysosome. All right, but what is believed to be the mechanism of action of this compound? So it's believed that titanosyne dichloride itself is actually a prodrug, and that makes sense. That compound is quite unstable, so you would expect that it would dissociate. And I'm checking just because I don't know if someone's telling me something. It's checking to see if so you have a dissociation, and the titanium is likely to be bound by something like transferrin, as we've already discussed. And so there were some indicators that the release of that metal will result in the transferrin uptake into the cellular environment, and then some sort of release into the cytosol. So this paper, well, not actually this paper summarizes this mechanism. So there was a very pivotal paper by Peter Sadler from the UK that suggests that perhaps something like ATP may be responsible as an ionophore to take the titanium from transferrin and to pull it out of the endosome. So that was the proposal there. That's not something that has been confirmed, but it's a promising possibility. And so then that can lead to the titanium getting localized in the nucleus for some amount of time, and then also being able to be delivered to the lysosome. Nonetheless, some interaction of this nature is what triggers then ultimately into apoptotic cell death. Okay, so it is proposed that titanium, when it interacts with DNA, that it binds to the phosphate backbone, and that will make it distinct from platinum, which, as we discussed, platinum can bind to the bases, the nitrogens of the bases, and typically is going to be on the guanine groups. But what it was once thought that perhaps because it can bind to the phosphoester groups, it's not actually phosphate, but the phosphoester groups at the backbone, then maybe the titanium induces phosphate hydrolysis, and so that would cleave the DNA. But more and more is believed to be the case that it's not actually cleaving the phosphodiester groups, but rather maybe the metal will just bind to the phosphate. So here is an example of a derivative of titanosyne dichloride. This is not actually titanosyne dichloride. It's a derivative that was that demonstrates superior activity in terms of its stability in water, but also its ability to attack cells. And in this model, this was proposed by Matthias Tack, the titanium will coordinate to the backbone of titanium while still retaining the cyclopentadienyl rings, right? That's not believed to be the case for titanosyne dichloride. It's believed that the ligand is completely dissociated. But this is an interesting proposal because then you can see how you can have an intact titanosyne moiety coordinating to the backbone, disrupting it without actually inducing any strand break. All right, so again, going to then what's attributing to the mechanism of action, cytotoxic mechanism of action. So for a long time, people were thinking it's transferrin. That is the key to the activity of this metal. And you will see for a lot of metals that transferrin is believed to be the delivery agent that facilitates activity. So this was probably, I would say, in the late 90s, early 2000s, people were thinking, okay, well, the key to developing an effective titanium drug would be making a compound that can deliver the titanium to transferrin. And that seems like a reasonable approach, because you do have the overexpression of the transferrin receptor, and that overexpression in cancer cells versus non-cancer cells would allow for some amount of specificity. And so I would say that this is probably true for how titanosyne dichloride behaves, given that that compound is not a very stable compound, and you would imagine that if you do bind a transferrin, you can effectively enable the metal to go within the cellular environment. Something that's interesting, but may not be a primary source of activity, is that titanium-bound transferrin can actually, at a high enough concentration, block iron-bound transferrin from getting into the cellular environment. Okay, but that's not actually believed to be a relevant thing because the amount of titanium that will be available in blood versus how much iron is available in blood would not believed to be high enough to be too much of a competitor there. It's also been observed that the compound titanium bound to serum transferrin with both of the metals in the two metal binding sites, you need a high concentration of that complex to even begin to see any sort of cytotoxicity or any sort of anti-proliferative behavior. So you need a really high concentration of this complex, which is not believed to be feasible in delivery into the body. But this behavior of really high concentration goes in line with the high requirement of titanium chloride to see any effect. So that's why I said that I do believe there's a correlation between titanium chloride and titanium delivery to transferrin. But in general, trying to use transferrin as an optimal route to take advantage of titanium for a cytotoxic effect is not something that's really feasible thing. And you can see how people have made different derivatives of titanium dichloride, and really things that have improved the stability of the metal, still enabling the metal to bind to transferrin, but really not a huge improvement in terms of IC50. You still have these derivatives. These were made by Enrique Melendez, UPR Miami West. These are still pretty poor in terms of the IC50 values. All right, and you can come across certainly in the literature other titanosyns that have even lower IC50 values, but even then, you look at them closely, it's probably because of a different mechanism in place that's facilitating their activity. All right, so transferrin can maybe enable something like titanosyne to be effective, but in general, not a really good route for exploiting that anti-cancer property. So also people consider, well, what about serum albumin? I've already mentioned to you that serum albumin is an important protein for really fine-tuning some of the how long certain drugs exist in the body. And so it was natural to look at, well, something like titanosyne chloride, can it interact with this protein? So I don't have any visuals here, but it has been observed that you can really stabilize titanosyne or titanium itself at really high equivalence of stoichiometry with the protein, but you can even form like a 1-1 protein compound complex. But even that complex, even as high as 100 micromolar concentration, does not have a cytotoxic effect. So certainly albumin may be able to stabilize the compound, but is not attributing to cytotoxicity. Interesting enough, though, Tact observed that his own version of titanosyne Y, which is basically one of his big contributions to the field, it didn't advance to clinical trials, but it certainly had a really interesting properties in that much more stable, much more effective in terms of potency in different cell lines. And it was observed that this compound shows a dependence on the presence of albumin to actually have an anti-proliferative effect. So with this version of the compound, and it may be a compound-dependent thing, it has to be associated with albumin, so some sort of interaction, some sort of binding interaction that allows the metal to be active. Maybe it helps it to be delivered into the cellular environment. All right. So over the years, people have really moved beyond just like the initial form of titanium in the titanosyne form. You have a lot of sort of like next generation titanosynes and even titanosynes that have been mixed with other ligands to try to bind it with other metals so that you have sort of bimetal compounds. But you also see people have pushed beyond that modality. So titanosyne, you're talking about an organometallic compound. So you have carbon-titanium bonds. But people have then looked at, well, what about titanium in terms of its hard metal properties binding to ligands that contain oxygen and nitrogen features? And so you would say, well, look back to butyrotitane, which was something that was oxygen-based, and it was also not any better than titanosyne, right? So it's very clear that there's going to be certain features about the ligands that have to enable it. So you had people like Shuva, who really pushed the field in terms of the types of ligands that you can look at, and the observation that you can have ligands that can really strongly stabilize the titanium, and that in the cellular environment, these compounds can be quite active. And so you have a move away from compounds that are very labile and active to compounds that are very stable, but that in the intracellular environment become active. And so I'm not going to go into much of this actual thing, only to point out that it does appear to be very compound dependent. The activity of the metal as maybe facilitated by the ligand. So some of these other compounds here may not actually operate how titanosin operates, where it's believed that the titanium will localize to the nucleus. It may be other things by which they operate. So I'm gonna touch upon some of this. And so just to emphasize, you do have a lot of these newer titanium compounds being more soluble, that's a plus. Being more stable to hydrolysis, that's a major plus, because oftentimes the ligands are important, maybe not for activity, but certainly for delivery of the metal, okay? And so that has led to compounds that are more anti-proliferative, cytotoxic. But also you have findings where you want a nice balance between stability and lability for the metal to be able to reach its cellular targets. And even the finding that you can have highly stable and inert. So stable is in terms of high affinity ligands to the metal, inert meaning that a poor ligand exchange, but that the lability of the compounds can be triggered within the cellular environment. So some work that I personally have done have shown that you can develop compounds with ion chelators to induce this lability by a transmethylation effect, where essentially the ligands are going to release the titanium and bind iron. So that's another route by which this can be facilitated. So here I'm going to show you an example. This was a collaboration done by two different people in the titanium field. This was done by Shuva and also by Matthias Tack. Matthias Tack was the person who developed titanosin Y, which is probably one of the most effective titanosines. And then Shuva, she's worked with saline type of ligands, okay? And so interestingly, how these two compounds behave. So what we're looking at here is the uptake of titanium in the cellular environment. So here is a cell line, the HT29 colon carcinoma, and we're looking at uptake as measured by the amount of titanium with respect to cellular proteins, or even just the concentration of titanium within the cellular environment. And so what we're observing is a time-dependent behavior. Another really important thing is treating the cells in the presence and absence of serum. So when I introduce to you cell work, I talk to you about how cells media is often supplemented with serum because of the proteins that are present that enable them to be able to grow effectively. Well, here the question was, can the serum influence the amount of metal getting inside the cell environment? So if you look at the titanium salin case, what we observe is that in the presence of serum, so these are the ones that are in black, okay, you see that you get higher, much higher uptake of titanium in the cellular environment. Whereas the absence of serum, you have sort of a much lower amount of uptake. But in the case of titanosin Y, you get the reverse trend. Although you have these high error bars, you observe that in the absence of serum, you have a higher uptake of the metal into the cellular environment, whereas in the presence of serum, it's something that's less. Okay, but that I'm talking to you in the level of uptake. I've mentioned to you before that doesn't necessarily go in line with activity. Okay, but I'm just showing you how serum can certainly affect the properties of the compound. And where is it coming from? Well, it's obviously coming from the ligand, what's surrounding the metal, and how that can enable interactions with proteins, and most likely in this case proteins, but even maybe some small molecules in terms of what's facilitating the uptake of the metal, or even maybe the metal compound into the cellular environment. All right, so this is an interesting study. So this was an extension of the work done by with Salin, and this was actually a compound that was developed by Hun, not by Shuva. So here, what was done in terms of the coordination chemistry was to use a saline group. So you see the coordination of the saline group, which is a tetradentate ligand, and then to incorporate a tridentate ligand. This was a dipic molecule to create something that's 7-coordinate. So interestingly enough, it's a very stable compound in terms of its behavior, its solution behavior in water. And it does have really broad spectrum effect. So you treat different cell lines, you will have cytotoxic properties. But then in this study, they were using titanium PET. So that's a technique that is an imaging technique to try to track the metal. And as it's employed in this particular mouse model. And so in this mouse model, they actually did a H29 xenograph. So they took a human tumor, a human cancer cells. They put it into the mouse, let it grow into a tumor, and that was positioned in the flank of the mouse. And so what we're looking at here is over time, okay, so 1 minute up to 7 hours, we're looking at the movement of the titanium through the actual mouse, okay? And so these different arrows are pointing to different organs within the mouse. So the red arrow may not be as easy to see. That's pointing to the heart. And then you have the orange arrow that's here, that's pointing to some amount of localization after 10 minutes. That's in the liver. And you have in the gallbladder at 30 minutes. And then you have some amount of it in 3 hours in the area that's called the cecum. And then at the end, 7 hours, you have an amount of it that's in the colon. So you see that it's getting excreted over 7 hours. What you don't see is much accumulation where, Alex? So where's the tumor? In the flank. The tumor was in the flank. But you never see an actual accumulation there. You see movement through the whole system, but never actually accumulates there. So compound, you test it with cell lines and those plates, you have something that's very active, but then here in this particular study, it didn't have much accumulation within tumor at all, and also didn't have much of an impact on the growth or decreasing the size of the tumor. All right, so in addition to targeting DNA, and as I was mentioning to you already, it's believed that the mechanism of, or the contribution of titanium to the mechanism of action may be compound dependent. So what else has been observed is that titanium has a capacity to inhibit a number of intracellular enzymes that may directly, and this inhibition may be direct, as in like coordination to the enzyme results in the enzyme losing its activity, or maybe something where the titanium binds to the substrate of the enzymes, and by that coordination makes those substrates not recognized by the enzyme. Okay, so that's something that has actually been suggested for something like ribonucleotide reductase. I mentioned that previously, which is an iron-dependent enzyme where the titanium might be able to bind to the nucleotide substrates. Another thing is that titanium might have the capacity to contribute to the inhibition of the bioavailability of the labile iron pool. So not directly affecting the pool, but with the use of certain chelators, affecting that pool and affecting how that metal might be able to activate important enzymes. And so again, I've kind of jumped the gun, this effect of inhibiting ribonucleotide reductase by affecting the nucleotide substrates and basically then making that enzyme not be able to exhibit this function. All right, so that's believed to be other proposed cellular targets. That's something that's still being studied because again, it's very compound dependent, the effects that that metal can have. All right, before we move on to gold, any questions with regards to what's believed to be attributed to the cytotoxic properties of titanium? Any doubts or any questions? No? See, so not necessarily for like an anti-cancer property, but there are certain strategies that people try to exploit to do with the magnetic properties of the metal, right? So you even have some studies where people look at like iron nanoparticles, which are magnetic, and they try to exploit that feature. But in terms of by itself, just being magnetic or diamagnetic, that's not going to influence. It has to be some sort of external tool that you're trying to exploit the actual magnetic property. Other questions? All right, so let's move on. All right, so we're going to look at now gold. So as I mentioned to you, this is now at the other end of the spectrum in terms of hard, soft at the base chemistry. Now we're looking at something that would be on the soft end. All right, so when we're looking at gold biologically, we're looking at the metal in terms of its +1 and +3 oxidation states. So I think, Jaylene, your lab works with gold nanoparticles, and those tend to be gold 0, as far as I understand. Ustedes trabajan con oro.
Sí, en particular
Sí, como con citratos.
y se modifica
So typically, you're going to see +1 and +3 when you're talking about small molecules. But again, some of these nanoparticles, I've seen them to be neutral form. So when we're talking about the reducing environment of cells, you're most likely going to see gold in this +1 form because it's favorable for the formation of that. Now, as far as the coordination chemistry, we have gold 1 being, so I'm going to focus on gold 1 first of all, have an electronic configuration of D10. All right, so that's now way different from titanium, which was D0. So D10, meaning that all those orbitals, the d orbitals, are full of electrons. So full of electrons, and if you're talking about an octahedral environment, which is probably not relevant for gold, but let's pretend it's relevant, what you're going to have is a 0 crystal field stabilization energy. Okay, and so because of a 0 crystal field stabilization energy, you're going to have compounds that are ligand exchange labile. So the structure of gold 1 complexes is determined by the steric effects of the ligand, so how big the ligands are, and how that will stabilize the coordination number, and also simple electrostatic interactions between the ligands and the metal ions, so the charge states of these. So here's a representation of the common coordination chemistry of gold 1. So it is a soft Lewis acid. You typically will see coordination number 2, 3, and 4. And this is what they would look like, as we talked about earlier in the course, some of the ideal geometries. And so this coordination number 2 is something that's preferred by anionic ligands, but also by ligands that are sterically pretty big to be able to stabilize that type of structure. And so as way of, the lability of the compound, here is just a representation of this exchange of phosphine groups and the formation of going from the coordination of 1 phosphine group all the way up to 4 phosphine groups to satisfy that coordination number 4 with respect to that particular type of ligand. So I'm mentioning this just to emphasize this idea of lability, how quickly there's an exchange. So something that's phosphine related, you can take advantage of P31 NMR. It's a very effective tool to be able to gauge these type of groups. The interesting thing that you observe, if you're looking at, let's say, 4 of these ligands bound to the gold, or even just 1 of these ligands bound to the gold, is that in the profiling this reaction, you don't normally see distinctive phosphorus peaks for the different species. Okay, so you don't normally see for this species versus this species, but you would expect to see it, right? Because you have different number of phosphine groups, and that has to do with the rate of the exchange, how rapidly exchange. You would typically not see this really distinct chemical shift in terms of how much of the ratio of gold to phosphorus you actually have. All right. Now, to contrast this a bit with gold 3. So gold 3, now you have the loss of 2 electrons. So you have an electron configuration of D8. So once you're at D8, we should start thinking already to what? What metal? To platinum. Platinum 2+, because platinum 2+ is D8. So these 2 metal ions are isoelectronic. And so now you're at D8, you're going to have something that would be square planar in terms of its geometry, coordination number 4. In terms of crystal field stabilization energy, gold 3 is more stable than platinum 2. And then you have things that are now ligand exchange inert relative to gold 1, which was quite labile. All right, so gold 3 is an intermediate metal, so now, or at least pushes towards intermediate, probably not truly to say intermediate, but it pushes towards that, and it can coordinate a diverse set of ligands. So you have, formation of coordination to thiol groups, and these are the sulfur-containing groups, nitrogen atoms of DNA bases. So you can maybe expect to see some similarities in terms of the coordination that you would observe for platinum 2+. Now, in terms of the mechanism of ligand exchange, you have something that's going to be an associative mechanism of ligand substitution like platinum 2. So I'm not going to go into the details of this. This is something that you would discuss in a traditional inorganic chemistry course, this idea of the associative mechanism of how you can have ligand exchange at the metal center. Now, in terms of the presence of these metals, so within the cellular environment, I said you would have a predominance of gold 1, but depending on the ligation of the gold 3, you might have sort of a mixture of both, of gold 1 and gold 3 within the biological context. All right, So let's consider gold in terms of its therapeutic application. So a lot of what was initially explored, and you can see the use of gold therapeutically dates back to even the BC time. So you have the Egyptians using it, you have the Chinese using it. And so as far as its evolution of the metal itself, a lot of it stemmed from the chemistry of gold-1 and identifying that it had the capacity to be something that had an antibacterial property, and also later to have an anti-arthritis property. And so these are some of the compounds. So you have gold cyanide, but also some of these compounds that show gold-1 with a thiol group coordinating to the, or sulfur group coordinating to the gold center. And these demonstrate this antibacterial properties, also this anti-arthritis property. And so gold thiolates were classified as anti-arthritic gold drugs. They form the classification of intramuscular gold therapy, a disease-modifying anti-rheumatic drug, or DMARD. And so in terms of the delivery of such compounds, weekly, monthly intramuscular injection with a dosage of about 50 milligrams of gold. And one of the main effects of these compounds is the toxicity that occurs in kidneys, so the nephrotoxicity. Today, gold in general might be used clinically, but it's very rare for it to be used clinically. So these are some examples of gold compounds that have been used, but a really famous one that people still study today, although again, not necessarily in the context of an anti-arthritis property, is this particular compound, which is aranafin. So aranafin has this composition where you have gold-1 coordinating to a sulfur group that's part of this thioglucose modality, and then you have the coordination to a phosphate, so this triethylphosphine group. So you have this linear coordination to the gold-1 center. And so it was a drug that is FDA approved for oral administration. And it has some improved properties relative to some of the other compounds that were used. So this was pushed into the drug market in 1985, although again, not actually prescribed more for this particular application. Less excretion via kidneys and the reduced gold retention in general. So what you see a lot today in terms of studying aranafin or aranafin derivatives is the repurposing of the drug. What other properties does this compound display that can be used therapeutically? So you come across a lot of literature where use of this drug for different types of applications. And so you even have, like, when COVID-19 hit, you certainly had people probing, can this actually have an effect? And you do see some studies where it did have a favorable effect, but not necessarily something that would prompt it to go further on the more of the intellectual curiosity. Right? So different studies and different extents of effect that the compound can have. Right. But let's look at it for the potential repurposing for an anti-cancer application. So it was observed that, and why people became curious about the anti-cancer application is the following. So these classic anti-cancer drugs, 6-mercaptopurine and cyclophosphamide, so these are not gold-containing, but these compounds demonstrate immunosuppressive and also anti-inflammatory properties, which were the same properties that gold compounds exhibited when they were being used for an arthritis application. So that then drew the curiosity about, well, could aranafin then have some sort of anti-cancer property? And it was actually observed that arthritic patients that were receiving gold therapy had statistically lower incidences of cancer. All right, so it suggests that it could have an anti-tumor effect. And so it was observed that the compound is anti-proliferative against a number of cell lines, and it has the capacity to, its interaction with serum proteins, it actually seems to be something that's does not increase, or it actually decreases the amount of cellular uptake that the metal can have. And so I'll talk about that in a moment when I show you a slide about its mechanism of action. But it's proposed that this compound will have an interaction with albumin and the coordination between the metal center and the protein. But characterizing its general anti-cancer properties, it was observed that across different tumor mice models, that it was quite active in an IP administration against the P388 leukemia. But in general, for different gold compounds, it was observed that this type of modality of gold coordinated to these type of structures that contain the sugar group and that contain phosphines were the ones that had the most activity. And so that's why people became quite interested, particularly in Ranafin, given that it was FDA approved and its toxicity was generally understood. So some of the mechanism that's attributed to the Ranafin, well, at submicromolar concentrations, Ranafin has the capacity to induce what is known as a mitochondrial permeability transition. All right. What does that really mean? And why is that even important? Well, if you disrupt the permeability of the mitochondria, what that can lead to is the release of an important protein called cytochrome C. So cytochrome C, you probably have heard of it in terms of the metabolism, and it contributes to the electron transport, but also has another property besides electron transport. It has a property of triggering apoptosis. So the release of cytochrome C from the mitochondria is part of the cell program death, where that protein has to be released in order for apoptosis to be triggered. And it's observed that Ranafin can do that, can cause for that release to occur. Another important property was the capacity to inhibit an enzyme called thioredoxin reductase. So here we see the IC50 value of its inhibition capability, reducing the activity by 50% at a concentration of 20 nanomolar. And so it's proposed that inhibition is probably occurring because of the existence of a seleno group on the cysteine at the active site of that enzyme. All right. So why is that even important? Well, selenium is actually a soft element. And so you have a site, or you have this modification of cysteine. Instead of having the sulfur group or the thiol group, you have the seleno group. That's going to be favorable for possibly being coordinated or coordinating to GOLD1, and that coordination event will block the enzymatic activity of that enzyme. So this is a proposed mechanism. It's a little hard to see here. We have the Ranafin that is believed to not actually enter the cell in an intact form. It's believed to be in a modified form where you have the sugar being dissociated, and what you get is gold phosphine entering into the cell environment. So I believe that maybe its interaction with albumin might facilitate this, but that part is not so clear. What is understood is that once it enters into the cell environment, this particular metal is going to affect the thioredoxin reductase system, both in the cytoplasm. So this is the interaction of the gold compound infecting that system in the cytoplasm, but also in the mitochondria, because there's two different systems of the thioredoxin reductase system. And so it's believed that if we have this gold binding to the selenium group of this enzyme, that's going to affect this redox control that these particular, these set of proteins enable. And what that then leads to is an upregulation of reactive oxygen species, which can trigger a number of down effects leading to cell death or apoptotic cell death. So believe that coordination of the metal to the selenium of these groups, or even to proteins related to these groups that have a sulfur group, is enough to offset that control of the reactive oxygen species and can lead to cell death. All right. But as I mentioned to you about the coordination chemistry of gold, it is GOLD1. It is something that's quite labile. And the liability is not necessarily a good thing, because it could lead to your compound completely falling apart even before it actually reaches into the cell environment. So people have tried to fine-tune the types of compounds in terms of their coordination chemistry to improve their stability. So here's just a representation of GOLD1 in terms of that linear coordination and how you can get this associative mechanism for ligand exchange, where one of the ligands that are bound to the gold ultimately gets replaced by this incoming ligand, this incoming nucleophile, and you get this nucleophilic substitution. So the goal of what people have been trying to focus on or have studied over the years is trying to decrease the liability. And so one approach is by use of chelators that can enable a slower dissociation of ligands, because again, you don't want to lose that liability. You want the metal to be able to reach a target, but you don't want this to occur before it even has the capability of reaching the target. So the use of chelators, now you see how you're altering the coordination number, And by way of these chelators, now you're going to get this sort of, this slow dissociation of the ligand by way of one part of it become dissociated and then possibly another part for it to be ultimately displaced. So that's believed to be a principal way by which you can improve the activity by decreasing the solubility. All right, so let's look at some of the compounds that people have explored to improve the cytotoxic properties of the metal. So here's an example. We have gold 1 and using this type of phosphine chelator. So you know how we talked about amine chelators, well, this is a phosphine version, and you generate this type of tetrahedral type of compound. So you have these two bidentate molecules poured into the metal center. And so it's been observed that this particular compound or family of compounds have the ability to be broad spectrum in terms of their effects. And they can definitely target different types of cancer cell lines and also even attack different tumor models, animal models. It's also been observed to be more stable in the presence of thyroid groups, so more stable in the plasma than when you have the, say, aranophen, those type of compounds. But you still have associated toxicities. You have acute cardiac, hepatic, and vascular toxicities. Now, in terms of mechanism of action, even though coordination chemistry is different, it is believed that still you do have that effect of the gold being able to trigger a mitochondrial dysfunction, and that you can assume will also lead to cytochrome C release and the triggering of apoptotic death. All right, so let's look at this particular study. So again, it's the same type of chemistry, these type of ligands that are phosphine-based. What we're looking at a bit is the difference in terms of backbone, okay, but also in terms of the R groups that are on the phosphine. So these are the R groups that were tested in this study. So what do we see here? All right, so here we're looking at the interaction of this these two compounds. So these two compounds being here, one with the 1, 2, 3 back, and then one with the two carbons that form the back of the actual phosphine chelator. All right, so we're testing them against the MDA MB468 breast cancer cell line. That is, I believe, the triple negative cell line. And then you're also testing against the human mammary epithelial cell. That is a non-cancer cell line. And what do we observe? Well, we observe that both of the compounds are able to do a dose response effect on the growth of the cell line. So it inhibits, and we get about an IC50. We were trying to probe this at about 1 micromolar for both compounds. Okay, but then if you look at the non-cancer cell line, we see that this one to the left, that compound doesn't have a dose response effect. So you basically have 100% cell growth or cell viability, but the other compound does have this dose response. So you can see that one of the compounds does achieve some amount of selectivity for the cancer cell line versus the non-cancer cell line. Then another thing was probed using the more effective compound of the two that was observed, or the family observed, that more effective one, was to look at the activity against the thyroredoxin reductase system. So we're looking at thyroredoxin activity and thyroredoxin reductase activity. And what do we observe? So looking at both cell lines, so black is the cancer cell line, white is the non-cancer cell line. So control, no gold treatment, we get that the activity of the enzyme, thyroredoxin enzyme is 100% active, and the reductase is also active. But then treatment of the compounds in the cancer cell line, we see that this activity of the thyroredoxin is significantly decreased, and even for the reductase, but not as decreased when you compare it to the non-cancer cell line. So there's a clear difference in terms of activity of those two enzymes by these compounds, which could be contributing to why there's a difference in terms of the selectivity of that compound against the cancer line versus the non-cancer one. All right, so it's proposed, very similar to what I proposed for, that I showed for aranophen, that this compound will go into the cell environment and have a very similar effect. In the cytoplasm, the gold with a slow dissociation of ligands will be able to bind to the selenium group or the sulfur groups of those enzymes, and then perturb their regulation of ROS. in the cytoplasm and also the mitochondria. So we believe that the mechanism of action is still very similar. And then further down the line, similar mechanism of action, and you have further effects, which we're not going to get into, but it can lead to what's believed to be apoptotic cell death. And so that's attributed to the gold itself and its softness interacting with the catalytic site of these enzymes. All right, what else has been explored? So in the last portion of this, I'm going to show you a little bit of work with carbines. So carbines are a different type of ligands. So here now we're looking at metal carbon bonds. And so we're looking at basically these type of carbines, these NHCs, which are electron rich, and they can form neutral sigma donor. There are neutral sigma donor ligands that can form a bond through the formation of through this particular carbon group right here. So you can alter the electronics by changing the nature of the groups that are on the actual ring. So playing around with these R groups to try to then affect the chemistry of the gold itself. So people were playing around with these NHCs to see what can happen in terms of the gold chemistry. Okay, so this is a move away from the phosphine groups. And so what was observed is that they can form very strong bonds with metals, and in some cases stronger than the phosphines could. All right, so what kind of compounds are we forming here? But now we're going back to the linear one, right? So we're not looking at the tetrahedral ones, we're looking at these type of compounds. But then the use of the R groups being very important, because now we're talking about the sterics. The use of the R groups can cause a blocking around the metal center to try to protect the metal center and to try to minimize the dissociation of these ligands. All right, so here we're just basically playing around with the actual substituents and the use of these carbines. All right, so what was observed? So looking at this family of compounds, right? So the R's are these groups right here, propyl, ethyl, methyl groups. There are the R groups. And what do we see? Well, we're looking at the MB231 breast cancer cell. It's a different cell line. And we see that all three of them seem to have a dose response against this particular cell line. And then in terms of the activity against the thyroreducin reductase and a similar enzyme, but not the same enzyme, the glutathione reductase, we see that there's a selectivity towards the thyroreducin reductase, but not against the glutathione reductase. So these compounds do show the capacity to be cytotoxic, probably not so impressive versus some of the early ones I looked at, but nonetheless, still show that activity. And it seems to be selective towards the thyroreducin reductase system. And so I'm going to end up right here. I believe that selectivity may be because the thyroreducin reductase has that seleno group, which the glutathione reductase does not. It has a thiol group. And maybe that difference is enough to favor gold binding to the seleno group as opposed to the thiol group. Okay, so I'm gonna end right here. We'll continue on with there's not really much left, but we'll just finish this in our next lecture. Okay? So next lecture is what? A seminar, I believe. All right then. I'll see everyone. Have a great weekend.